This is the qualitative analysis and lead contamination presentation. For the qualitative analysis, group 1 cations of silver 1, mercury 1, and lead 2 cations are used. They are mixed and separated through different means in order to determine what is in the solution through visual observation. For the initial reaction, hydrochloric acid is mixed with the solution to separate the ions. For example, silver and hydrochloric acid create silver chloride and a hydrogen byproduct. Mercury chloride is not soluble, but mercury 2 chlorine 2 is. Lead 2 chloride is soluble, while silver chloride and mercury chloride are not. Silver chloride is soluble in dilute aqueous ammonia, forming a colorless diamine silver complex. For the second reaction, lead ions can be identified by using potassium iodide and potassium chromate solution. Silver ions may interfere with the potassium iodine test or the lead 2 ions if the procedure is not carried out properly, since both lead iodine and silver iodine form yellow precipitates. The potassium chromate test is more specific. It forms a yellow precipitate with lead, and the other ions will not interfere with the color. The reactions for the confirmatory test for the lines are lead 2 plus chromate creates lead chromate, and lead 2 plus iodine creates lead 2 iodine. For the final reactions, mercury 1 chloride reacts with aqueous ammonia, forming a black to gray precipitate of mercury and mercury ammonium chloride, which is white. The mercury ion is confirmed by the reaction. The silver ion can be confirmed through the formation of a white precipitate when the silver complex formed in the first reaction is acidified with nitric acid.
Lead contamination. How does lead contaminate? Lead contaminates the environment through both natural and human-made sources. Natural sources. Natural contamination of lead includes weathering of rocks containing lead minerals like galena, lead 2 sulfide. The latter version is PBS. Anglesite, lead 2 sulfide, PbSO4. Minium, which would be Pb2PbO4, aka red lead. Cerussite, which is lead 2 carbonate, or PbCO3. And volcanic activity. Volcanic activity introduces minerals into the atmosphere and surrounding environment. Now, human-made contamination includes lead-based paints, including like paints that you would use on your buildings or the roads, etc. Lead pipes and plumbing, which is a major source of water contamination. Gasoline additives, which is tetrathyl lead, which is phased out in many countries, but still relevant historically. Another one is industrial emissions. Smelting and burning lead releases lead particles into the air, into the atmosphere and ozone layer. And electronic waste, which would be improper recycling, which leads to lead being released into soil and water. Harmful forms of lead. These chemicals are harmful when ingested. Lead acetate, lead nitrate, lead chloride, tehydral lead, which is used in gasoline, highly toxic and can cross the blood-brain barrier. Lead carbonate, historically used in paints and can be ingested via dust. How does lead affect the body? Lead ions disrupt many biological processes and body systems. Lead ions disrupt the following processes: the nervous system and the hematopoietic system. The nervous system is disrupted because lead does something kind of strange to the neurotransmitters and how they're released. It impairs brain development, especially in children, and can cause cognitive deficiencies, memory loss, and behavioral changes. there's an episode of Law and Order that showcases the harmful effects of lead and what it can do to a child and an adult body. The hemiotepic system interferes with the hemosynthesis by inhibiting enzymes and chemicals, leading to anemia. Lead effects continued. The effect that lead has on bones can replace calcium and gets stored in the bone and serves as a long-term source of internal exposure. It reduces fertility in men and can cause miscarriage and premature birth. It disrupts the processes involved in DNA replication. It also disrupts nutrients uptake and waste removal and damages kidneys, leading to neuropathy. Lead reactions and instrumentation used for lead detection. Lead reactions. So the lead reacting with SO4 turning into PbSO4, it was a white precipitate. Then Pb reacting with iodine caused a bright yellow precipitate. Pb plus CrO4 turning into PbCrO4 caused a yellow precipitate. Complex formation. Pb can form complex formations with EDTA or crown ethers, useful in titrations or selective sensors. Instrumentation needed. Atomic absorption spectroscopy, AAS, sensitive and specific for Pb detection in blood or water. Inductively coupled plasma mass spectrometer, highly sensitive and can detect trace amounts of lead. X-ray fluorescence, used for detecting lead in paint and soil and consumer products. Anodic stripping voltammeter, which is electrochemical techniques useful for field detection in water. Advances in lead detection. Recent advances in lead detection technology strive to detect fainter and fainter traces of any possible lead contamination in water sources, soil, and manufactured goods. Colorimetric sensors use materials that change color in the presence of lead. For example, gold or silver nanoparticles functionalize with the specific ligands aggregated upon lead binding and shifting its color. Researchers at the University of Mazandaran in Iran have developed a new sensor that can rapidly detect the presence of lead ions in water. The colorimeter exhibits a high binding affinity for lead ions and results in a visible color change from blue to pink when binding occurs. Lead researcher Mohammed T explains that the new sensor has a simple and rapid detection process, making it ideal for field applications. The research team hopes that the new sensor will have a significant impact on environmental monitoring and water safety. The purpose is essentially just really trying their best to crack down on detecting lead in the most efficient way possible. G1 cations. These are essentially the ions, the ion reaction, the second reaction, third reaction, fourth reaction, fifth reaction at the end. And here are the pictures to go with it. You can read that on your own. There's no need for me to say it to you. And here are our sources. Thank you so much for your time. I hope that you somewhat enjoyed our presentation. Thank you very much.